Thanks all for persisting late in the day and halfway through the conference. Probably a little blurry now from all these slides. Thanks to ARM for putting this together. I think for me, sitting through this day, if there's a theme, it's that perhaps our field is now moving from exciting science experiments, which may get you on 60 Minutes, and our founder was there several years ago, but now to real products. And I think that's what we need to do to get the enthusiasm of investors as well as potential partners. As Jason said, we're in the clinic with, and probably best known for our urinary product applications, and we did make a presentation earlier this week that's been updated on our website. So if you want the whole 10-gen story, go to the website. What I thought I would do today is drill specifically into the promise of this renal program, as it is classically one of those multiple effect cell therapy products that potentially addresses a really enormous healthcare burden. So that's kind of my theme. You, of course, are here to listen to forward-looking statements. Where are we taking this in the future? So briefly, how our platform and how we think about product development may be a little bit different from some of the other companies. And that is to say that we don't have a particular cell type or type of biomaterials for which we are searching for applications. The founding premise of our company is that the human body has some regenerative capacity. You just have to give it the right catalyst. So what we try to do in each therapeutic product that we've developed is think about what cells or conditions are necessary for regeneration of an organ or a tissue. This may or may not be from the target organ. In the case of the cells, our urologic products are from smooth muscle cells derived from fat tissue, not even from the target organ. This renal program that I'm talking about uses cells from the patient's kidney. The biomaterials, in the case of the hollow organs such as vessels or bladders, we use a biodegradable scaffold for structure. In the renal program, we don't do that. We're not growing a kidney, but we use a gel-based formulation that gives properties that enhance stability and delivery, and so it is the cells with that biomaterial that are implanted into the patient. Jason mentioned the surgical implantation. You're doing something like bladder cancer surgery. That's a very involved surgical procedure. Something that's not going to work for somebody with chronic kidney disease. So we work with device developers to come up with an efficient, clinically acceptable way to administer this product into the kidney without requiring an open surgery. And finally, I think often unrecognized but really important for the for commercializing these products is, can you do this in an industrialized way with attractive COGS, with a turnaround time that fits the clinical parameters? When our founding scientist was first doing this, it might take 8-12 weeks. That was never going to work in a setting of bladder cancer where you had 4 weeks to get that patient treated. So with both the urology products as well as the renal program, we're talking about a 4-week turnaround for this autologous product that fits into what's needed clinically. So in a nutshell, I think every presentation today has talked about the enormous healthcare burden. Chronic kidney disease, this is one of the biggest Medicare spends in the country, $39 billion a year in annual costs. This is actually a relatively small population if you think about those dollars. It's about 500,000 people who've either had a transplant or on dialysis. It's expected to double from 2010 to 2020, so that's driven by obesity, hypertension, particularly diabetes, in an aging population. So this is one of the many healthcare settings that we talk about bankrupting the national budget. What we're trying to do then is say, okay, how can we intervene in a patient that's headed down that path before they get to dialysis and try to prevent or delay it? So in a simple overview, a routine kidney biopsy, we extract cells that are then isolated, expanded, cultured, selected, formulated into a prefilled syringe, and then delivered to the clinician for direct injection into the kidney in about a 4-week period. All right, so I'm going to drill deep a little bit on some newer data that have been submitted for publication that shows where this product is and really how we got there. So a little busy slide, but if you'll just indulge me, we started with, again, go back to our model. What does the kidney need? How does it regenerate? So we started in the first model with a 5/6 nephrectomized animal. Remove one kidney, stabilizes, remove 2/3 of the remaining kidney. This rodent eventually develops chronic kidney disease. So we wait until the animal begins to decline, and then we intervene. So several treatment groups. This is a Kaplan-Meier survival curve. First one I want to show you is the black one here. This is nephrectomized. You nephrectomize the animal, they're all dead within 4 months. Unfractionated means we took the cells, expanded them, and just boosted them back in. So you gave the kidney more of everything that it has, and it was no different than no treatment at all. So clearly it was the selection of the right cells in that formulation that would drive the difference. Another one we called B4, which is another band as we were testing very sequentially different combinations. We showed some boost. And finally, the one we're taking forward internally was B2, was the same as the sham nephrectomy. So after 6 months, these animals not only reverted their kidney disease, but they actually lived to the full natural lifespan of the untreated animals. So we said, all right, something real is going on here. If you look here, this is looking at creatinine, so one measure of kidney function, nephrectomized animals, creatinine increases. The line stops because all the animals are dead. Unfractionated B4, B2, we started to approximate, again, the untreated animals. Interestingly, when we looked at a whole organism effect, of course beyond survival, but weight gain. So these are still healthy animals. The sham animals are still gaining weight 30% during the last 6 months of their life. Nephrectomized and untreated animals begin to waste away, but we saw these animals actually gaining weight. So there's clearly an effect that was more than just one compartment of the kidney. The overall health of the animal is increasing, and obviously they're surviving. So the question, because we showed this at J.P. Morgan a couple years ago, and people said, oh, that's exciting, but it's a rat and it was healthy to begin with. So would this work in a natural disease model? All right, so this is the ZSF1 rat. Great model for this, I won't say uniquely American, but quite prevalent condition in the U.S. This is an animal that probably watches a lot of TV and eats a lot of fast food. By 8 weeks of life, it's developed type 2 diabetes. So it's an animal model, you may be familiar with it. It's a leptin receptor deficient animal. It doesn't sense satiation. It continues to eat itself to death, basically. So within 8 weeks, it has diabetes, begins to show renal nephropathy, only lives about a year, and has all these comorbid conditions, obesity, diabetes, hypertension, a very rigorous test of a therapeutic effect for our product. So we treated these animals at about 18 weeks. So again, they begin to get sick at 8. You wait until they've lost about a third of their kidney function already, and then we intervene to see, in the context of this severely diseased animal, could this treatment with a selection of their kidney cells make a difference? So the big takeaway is here's the survival curve. So this is out at 200 days. So this is about 3 months after the treatment began, and the blue line here is the obese untreated animal. Remember, their life expectancy is about a year, so this is 1 year of life. One died here. At about a year, they start to tick off. Top line, there's actually 2 control groups in the study. One is the lean litter mates, so that's the animals that don't have this genetic defect and they live their normal life. And those treated with our Neo-Kidney Augment, that's the NK treatment, survived well out past a year. So we're able to extend survival in these severely diseased animals. Really small numbers here, but here's the gestalt takeaway that was exciting to us is we saw impacts on virtually every parameter of kidney function. So we saw survival, we saw weight gain, we saw effects here in filtration, like creatinine, cholesterol, protein. In the tubule functions, sodium, calcium, phosphate, the ability in the loop of Henle to concentrate urine is measured by osmolality and specific gravity. Across all these various parameters, we saw improvement. So clearly more was going on here than just, you've injected its cells and it's boosted the kidney. We're actually changing the course of disease in these animals. In addition to the specific renal parameters, weight gain, whole effects on blood pressure, really quite exciting stuff. The next step, I should mention all this data on this diabetic animal model has recently been submitted for a publication, so there'll be a lot more coming out in that paper that also includes pathology, shows effects on fibrosis, inflammation. These animals, by the time they have a very sclerotic kidney and looks completely different when you look at the treated animal. Now, just to give you a little bit of preview of, right, so how does this start to look like a product as you move it forward? So next question is, well, what about a large mammal? So we did a similar study in nephrectomized dogs, and just to show a couple highlights, here's an improvement in GFR. This is looking at whole organ composites. We took each of those 3 kidney compartments and basically normalized the data in each one so we could combine the parameters and showed what does that look like over the course of the trial. And we see the untreated animals here just completely decline out to about 36 weeks when they begin to die, but again, a very significant bump with the treated animals. Similarly, improvements in weight gain and protein balance. As a quick summary, to keep us on time, I've mentioned the first 3. Not shown here, the question is, well, would this work with human kidney cells? We have shown that we can isolate, characterize, and expand these same cells from a severely diseased human kidney, and to do, a proxy for a clinical test, as a start, we took those cells into a nude rat model, an animal model that won't reject the cells, and showed that even in a chronic kidney disease model in that animal, the human cells showed a similar effect. So having now tested this across all these different animals and had preliminary discussions with the FDA, where we are now is we are targeting, we've said publicly, a pre-IND filing in the next few months, so first half of this year. I'll say by history, we've had a great relationship with the FDA, and great, not meaning they like us, but we've had the same review team for 5 years. Our prior two NDAs were accepted within 30 days based on 3-month IND programs, or GLP programs. So I can't predict, and I won't get too forward-looking on what they're going to do in this particular case, but we're excited that we'll be able to pull all this data together to go with them really in the next couple of months to say, what do we need to do to advance this product to human trials, because clearly there's a need here where a product, if we can replicate these effects in humans, would be a huge advantage. So with that, I'll wrap up. You want questions, or do you want to go on?
I'd like to just focus you on a couple of questions.
Sure.
So this is autologous, yes?
Yes.
And this is, so if I ask what is it, you're taking cells, epithelial cells from the kidney itself?
Yeah, so we're taking a routine needle biopsy and isolating, expanding and isolating what we're putting in are largely renal epithelial cells, not entirely, but mostly. We sort of stumbled into this away empirically, as I showed early on. We kind of tested very different combinations and even using the whole gamish of cells. And along the way, we ran into Ben Humphreys from the Harvard Stem Cell Institute, who had done work showing that it's actually renal epithelial cells that are the primary source of injury recovery in acute disease. And so that kind of validated in his model the same kind of approach for the cells that we were using.
Okay. When you have the cells expanded, what would the release criteria look like on the product itself? Have you characterized, so it's going to be a heterogeneous cell population of epithelial cells, or will it be a more tightly controlled homogeneous cell population?
Yeah, so it's a good question. We're working through the potency assay issues for the IND. So what I'll tell you is that it is an admixture, though it's primarily these renal epithelial cells. We've used markers to characterize that we're getting the right cells in the right proportions. How far beyond that we need to go with an autologous product, which in our experience has been not as onerous as maybe some of the allogeneic models, we don't know yet. In the case of our urology products, this is a closed system manufacturing. You can't do destructive testing. So we really are able to use markers of cell activity just by testing the media to establish release.
Thank you. One question that I have is, why, if I am extracting epithelial cells, expanding them and putting them back in, what's the mechanism of action by which they're repairing the kidney? I mean,
why is this happening?
Yeah, why is this happening? Because these cells are already resident in the kidney, so what I'm doing is I'm putting in, I guess, a super dose back.
Yeah, that's right. I think what you're doing is boosting the balance within that kidney. So we saw even in the most severely diseased kidneys, and we have an arrangement with UNC in Chapel Hill, and they'll give us cadaverous kidneys from people who have been non-compliant on dialysis for years. I mean, this kidney looks like a walnut. Even in that really diseased kidney that's entirely shot through with fibrosis, there are still healthy cells in it. In fact, these epithelial cells are upregulated because the kidney's trying to work. The cells are present, but the pathways are shot. So in terms of the effects, we showed in the 5/6 nephrectomy model, which has been published, that we've got cells lasting at least 6 months. So we believe there's certainly engraftment. And again, this ties to the notion of the epithelial cells being the primary repair cell. And we also think we're seeing paracrine effects from these cells. In the diabetic model, we showed we didn't do the same long-term study, but we showed 24-hour testing with with tagged cells. So it's not just a matter of you boost something in this kidney and they all squirt out. They actually stick around. But we think it's both paracrine and engraftment and repair.
Okay. Thank you so much. Maybe we'll have a chance to come back at the end and